Thank you so much, Ed, for that kind introduction, and thank you to the FDMAS Alliance and the International Consortium for inviting us here today. So Dr. Fernando Fierro and I will be sort of joint sharing this talk, and we'll present today our preliminary findings on our project applying single-cell transcriptomics to understand fibrous dysplasia of the bone. And this disease, again, needs no introduction to this crowd, but just a reminder that FDMAS really exists along a disease spectrum. So, we have patients that we take care of who have one bone that's involved, and other patients who have polyostotic fibrous dysplasia. And then on the more severe spectrum, patients who have McCune-Albright syndrome and exhibit multiple of the extraskeletal manifestations as well. And I think it's important to keep in mind this disease spectrum and this phenotypic spectrum when we're thinking about this disease, because this really, our animal models that we have, while they do recapitulate this disease, they can't take into account this phenotypic difference. And so I think using human samples and human data becomes really important. I think we can gain a lot of potential insights, and it can also be used to help improve our animal models as well. And the reason that there is this phenotypic heterogeneity is because the GNAS mutation, which is the causative mutation in fibrous dysplasia, it typically occurs early in development. So depending on when the mutation is acquired, that can lead to either a less or a more severe phenotype. And this figure is taken from Dr. Boyce and Dr. Collins' wonderful review in Endocrine Reviews from a couple years ago. So when GNAS is overactivated in the bone, that's what leads to the fibrosis and the abnormal trabecular bone formation. And the sort of longstanding paradigm has been that there is a skeletal stem cell or osteogenic precursors that carry the GNAS mutation, and because of that have altered signaling in the G protein coupled receptor signaling pathway, and that there is preferentially a block in differentiation so that precursor cells, these fibroblastic precursor cells build up, and you have this block in differentiation into mature osteoblasts. And this occurs at the expense of other cell lineages, and really is what we think is what contributes to the pathophysiology in the bone. But there are still a lot of questions that we don't understand yet about fibrous dysplasia. What specific cell types carry the GNAS mutation? And which cell types are responsible for the pathophysiology within the bone lesions itself? And then what signaling pathways are dysregulated in response to the GNAS overactivation? And single-cell RNA sequencing is a wonderful tool to try to sort out some of these questions. It's been used heavily in other fibrotic diseases as well. And so what we have been doing is performing single-cell RNA sequencing on human fibrous dysplastic bone lesions to try to answer some of these questions. So very briefly, we have been fortunate enough to collect bone samples from a handful of patients with fibrous dysplasia. And because this is a rare disease and most patients don't go to surgery, we didn't have any inclusion or exclusion criteria. Any patient who was willing to provide us with the portion of the bone that was not needed to make a pathological diagnosis, we could collect that. And so as you can see here, I just want to point out that there's significant heterogeneity in the samples that we collected. There's a pretty even distribution of male and female sex, but the location of the bone varies between long bone involvement or craniofacial involvement. And there is a wide spectrum in terms of the ages of patients that we've collected from, which potentially could impact the interpretation of the results. In order to understand differences between fibrous dysplasia and then the cells from healthy bone, we had to think about what type of control samples to use. And healthy patients are not going to get bone biopsies. So what we chose was patients who had osteoarthritis, who were undergoing joint replacement. They take out a good portion of healthy bone that's sort of distal to the replacement site. And this is what we used for our control samples. And today we'll be presenting the results on the long bone samples.
So very briefly, the method to isolate the cells. So the idea is quite simple, right? We said we want to see the different cell types individually, one cell at a time, And how are they different in FD versus normal cells? So how do we get to make single cells out of our bone sample? First, mortar and pestle, like witches, doing the old school grinding. Then we use collagenase to break the cells. This procedure is very important to clarify, because if it would have been different, perhaps, for example, in our final samples, we don't see osteocytes probably because they use a lot more collagenase before you can get to them, and EDTA and so on. But then you start to affect the viability of other cell types. So we had to come up with a compromise to see as many possible cell types. Then we select for the living cells, because of course some cells could have died from it's been at least 2 hours since they were resected from the surgery until we are really at that step. And then when you look at this mix of bone and marrow, it still looks, a lot of it is hematopoiesis. It's still from the hematopoietic lineage. And that makes so much of, for example, in the bone marrow, it's over 90% for sure. To enrich for the skeletal cells, the stroma, that component, we also did a selection trying to remove all the CD45 positive cells, which is a common marker for all the hematopoiesis except for the red blood cells, but we got rid of the red blood cells using a density gradient. It's a different procedure. So we actually have both samples collected. We have what we call CD45 pre, means the whole sample that contains osteoclasts and so on. We know they are there, but we haven't analyzed those carefully. We're gonna now just focus on what we see in the CD45 negative fraction, where we could really zoom into those cells that have more to do with bone, cartilage, stroma, and so on. I'll show you. So very briefly, we don't need to go into the details, but why did we not do this 20 years ago? It's because we didn't have this technology even 10 years ago. And basically what it does is that you can load the single cell suspension that I just showed you how we did into a chip where it goes into a microfluidic system where each individual cell gets encapsulated, and you can extract the RNA, reverse transcribe it, and then sequence that. And it goes with a barcode, so you know exactly what cell were you interrogating in the first place and what genes do they express. It's amazing technology. And we see all the genes that are expressed if you have enough depth, right? So it's often, how many, we have 20,000 genes maybe that code for protein, something like that. There is many thousands of genes that we will see. And this is how they look. The UMA plot is a very common way of representing single cell data. It stands for uniform, manifold, approximation, and what's the projection. And all it really does is to cluster the cells by similarity in gene expression, right? So two cells that have identical genes at mRNA levels of all what we were measuring would cluster very close to each other. If they're very far from each other, it's because they have very different gene expression. And so the first thing that we notice, I'm sorry I don't have pointer, but if you look at the middle panel, you can see that the clusters that we see that hopefully would have represented cell types can be roughly first summarized into 4 main clusters. One that we want to call the stroma cluster, and we're going to zoom into that because that's, of course, our most interesting one, has the skeletal stem cells, the osteoblasts, and so on. There are endothelial cells, there are some arterial endothelial cells and venous endothelial cells. There is something that is myogenic. We're calling it smooth muscle-related cells. And some cells are just really pulling apart because they are cycling, because during cell division, you will change a lot your genes that have to do just with proliferation. And those also cluster separately from the other 4. And you can see that when we do this, this is an integrated object. So it means that we took all the transcriptome from the 4 FD samples, the 4 control samples, and pulled them all together to get like a summary picture, basically. And you can see that all samples contribute to these main clusters. All these cell types, general picture was seen in all of them. So, and this is just a way to argue why we think which is which. One is, for example, to look at a PECAM1 in the middle. You can see it's a marker for endothelial cells, and you see it's highly expressed in what we call, the top panel and the bottom panels are still the same cells. They're just in a graph, they were like reorganized. But the But the endothelial cells, believe me, are those expressing an endothelial marker. Stromal cells express collagen 1, for example. It's pretty much expressed in all of them. And the muscle cells have also muscle markers. And this is even more detailed because each of these groups will have not 1 or 2 genes differences, right? They are typically between 100 and 1,000 genes that are different. And that is what we use to define who is who, what are their cell types. I should clarify that cell type should always be strictly defined by function. That's what defines it. We have 200 plus cell types in our body. It's because of what they do that we call them a different cell type. But we are using as a surrogate what they express and use that as markers to tell what they are.
So one of the benefits of combining everything into an integrated object is that we can perform differential gene expression, which means that we can look at the transcriptome of the fibrous dysplasia samples, cells in the fibrous dysplasia samples, compared to the control samples. And when we do that, we can do that for each of these individual groups, and we get this list of genes that are differentially expressed. And that is on the order of thousands of genes, so it can be challenging to just look at them on their own. But there are many software programs and tools that can be used that can look at how these genes that are different in differentially expressed in fibrous dysplasia versus the control samples, how they affect different biological pathways, which when we're talking about cell function, that becomes a very important question. And this is using ingenuity pathway analysis, which is a QIAGEN product. But you can see very generally that there are very similar upregulation of pathways in the fibrous dysplasia samples in each of these major groups compared to the control samples, which is something that's very interesting. Orange means that these pathways are significantly upregulated. And so this is just a list of 15 or 20 of many different pathways that are upregulated.
So now we zoom into the stromal cells, what I was mentioning before. So we break this down, then I should clarify, again, I said the different dots, basically each dot in this plot is a single cell, right? So we're looking at a couple of thousands of cells at once. This is the sum of all the wild-type NFT cells. And now we are using, there is one part that is a little bit arbitrary when you use single-cell RNA-seq that has to do with dimensionality and resolution. If you go high enough in dimensionality and resolution, you should find out every single cell is different from another cell. No cell will be identical in the gene expression to another cell. But we find a compromise where things make sense in terms of defining groups that have common markers that are typically associated with that cell type. And that's what we do with the stromal compartment now isolated from the endothelial cells on, so that we can not have that as a reference, endothelial cells or the muscle cells are basically too far from the stromal compartment. We remove them from the analysis so that we can really zoom in into the differences that we find just within the stromal cells. And first, I think it's interesting to notice that the clusters overall still overlap very well between wild-type and FD, but not entirely. So again, we're gonna miss the pointer, but there is in the bottom, for example, there's a big cluster 7 there. That cluster is only in FD samples present. It's a cell type that we don't see in healthy volunteers, only FD lesions. Similarly, if you look at the orange top left cluster 1, you see that that's mostly red only. It means it's a type of cell type that we only see in FD. And there is one that is exactly the opposite, cluster 3. It makes like an island on the east side. You see it, it's a big kind of brown-green cluster. That's a cluster that we actually only found in the healthy samples. And we think that that cluster actually has something to do with, I will show you now, with the anatomical location difference that we have between the FD lesions and the control samples. Here are the different clusters. It's very complicated. Actually, just looking at normal bone was something that I'm realizing most people don't have completely figured out. We had overly simplified this. Of course, we have our osteoblasts that we expected to see. We expected to find chondrocytes. They are there. But there are various groups of types of fibroblasts that I still wonder if they are really different cell types or just cells that are exposed, for example, to slightly different environmental cues and changing their gene expression based on that. For example, there is one fibroblast cell type that clearly has a lot of interferon gene responses. So maybe has something more to do with where they were at the moment of the resection than of being a particular cell type. Again, we have the proliferating cells, and so on. And this is, of course, I don't expect you to memorize this. It's just looking at what was a little bit of the criteria, some of the genes we were using to say, yeah, that's why we call them. For example, that cluster 3 I was mentioning to you that is only in the healthy samples, well, they actually express typical markers for synovial fibroblasts. And the synovium is in joints, right? It's right over there. So perhaps that's why we could never find them in the FD samples. The 2 clusters that are unique to FD, I think, are both very interesting. The cluster 1 is some type of fibroblast that is actually very similar to what is cluster 0. So perhaps it's still a fairly normal cell type, fibroblast type, that changed in sufficiently gene expression just to be looking different. And cluster 7 that you see, it's very far from all the others. It's an extremely interesting cluster because it actually co-expresses fibroblastic marker and osteoblastic markers. So it seems to be some kind of osteoblast, but really still expressing fibronectin and things like that. So it's the typical fibro-osteo cluster that we need to interrogate with much more detail to understand where do those cells come from, for example. That's a type of analysis we still have to do. And this is just like a summary figure of that. I'm basically saying there are mostly various colors and flavors of fibroblast. There are what we call skeletal stem cells or progenitor cells. They are in both samples. There are osteoblasts, chondrocytes, and the proliferating cells.
And once again, we were able to perform pathway analysis on the different subclusters here, where we could look at the fibrous dysplasia versus the control cells. Again, this is just a broad overview, but I'd just like to point out that we see upregulation of pathways involved in fibrosis, which is not surprising, as well as cell growth, differentiation, cell-cell adhesion. So this really is ripe for further exploration. And just moving on to our conclusions, so the non-hematopoietic or the CD45 negative compartment of the bone and bone marrow, we've shown contains these myogenic type cells, endothelial cells, and stroma. And the stroma appears to be comprised of these various types of fibroblasts, osteoblasts, chondrocytes, and these stem or progenitor cells. The FD samples did not contain one of the cluster that was present in the control samples, which may be these fibroblast-like synoviocytes, which may be due to differences in anatomical location, although we need to explore that further to see if this is actually a biological difference. And our FD samples contained 2 unique cell types that were present in FD and not control. And our future studies will elucidate changes. We're going to look at the hematopoietic compartment now, including the osteoclasts, and we also need to do the craniofacial lesions as well. Okay, and thank you. These are just our acknowledgements. This work was done, couldn't have been done without the MDBR grant funding supported us, as well as the NIH funding as well. And thank you to Dr. Ed Shao as well, who is instrumental in leading this project.
All right, well, thank you very much. Do we have time for questions? No? Yeah, okay. One question. Okay, so we have some time for one question.
Thank you very much for the presentation. I was just wondering if there is any way to understand which and how much of those FD cells you isolated do harbor the mutation.
So we are in the process of doing that analysis. We do have a technique for determining GNAS positive or negative cells, and that is the next step in the analysis to see if there is a difference between the cells that carry the GNAS mutation and those that do not. It's a great question.
Thank you.